Hi friends, please do subscribe and keep liking my videos. Your one click really a lot to me. In this video, I will discuss about enzyme linked receptors and their some other names and how they work. So let's try to understand why these receptors are called as enzyme linked receptors or transmembrane enzyme linked receptors. It is very simple. As you can see, the effector domain of these receptors are enzyme and they are tyrosine. In the image you can see RTK is mentioned, which stands for Receptor Tyrosine Kinase. I hope that you guys are very well aware of about enzyme. The basic definition of enzymes are that they are made of proteins and they act as biological catalyst. And catalysts accelerate or increase rate of chemical reactions. Now come to the point we are discussing about the enzyme linked receptors. Other way we can say that these receptors are attached with enzyme. That's why their name is enzyme linked receptors. They are also called as transmembrane enzyme linked receptors because these receptors lie across the cell membrane. Hope these two terms I discussed about enzyme linked receptors are clear. So far you have learned why these receptors are called enzyme linked receptors or transmembrane enzyme linked receptors. One more term used for these receptors I want to let you know, that they are also called as one pass receptors. Can you tell why so? Yes, they pass through cell membrane only once, unlike G protein coupled receptors. As you know that G protein coupled receptors passes cell membrane 7 times. The other important point is to understand the structure of enzyme linked receptor. Basically they are composed of 3 key domains. The first part is ligand binding domain. Here you can see, as name suggests, it is place where ligand like hormone, peptides get bind to receptor, and this part is lies in extracellular region. And the second domain is called transmembrane domain, which is composed of hydrophobic amino acids, and this portion of receptor attached to the cell membrane. The third part of receptor, which lies intracellular region and is also known as active enzyme domain. The majority of active enzyme domains are kinase which phosphorylate the amino acids. The whole process from ligand bind with receptors and activate receptors and then send signal to intracellular domain is also known as signal transduction by enzyme linked receptors. Here I am trying to make you understand about the steps involved in signal transduction by enzyme linked receptors. At the very first step, ligand like hormones bind with receptors at binding site or binding domain, which leads to dimerize or to make dimer form of 2 neighboring receptors which are previously inactive and lies in state of monomeric helical form. After binding with ligands, 2 neighboring receptors come closer, like in this image, and get autophosphorylated. Phosphorylation means adding phosphate molecule, for example converting ADP to ATP. And this phenomenon, the autophosphorylation, triggers phosphorylation of intracellular protein, which changes the shape of protein, which is also called as conformational changes. And this rapid phosphorylation leads or generate the response. Here you can note down the types of enzyme linked receptors. They are basically 5 types. Most important one is receptor tyrosine kinase, about which I have talked in this video. And thanks for watching.